Welcome back everyone. This session is titled Basic Research 1. Our chairs for these sessions are Jin Han and Derek House-Roy and Youngun An. We have 4 speakers presenting in this session, and a Q&A session will follow after the presentation. Now, I'd like to hand the session over to the chairs to introduce the panel members and proceed with the session. Chairs, please take over.
Okay, thank you. So it's my great pleasure to share with this session as a chairperson. And then my co-chairperson is actually mentioned that the Derek, House and Roy, and the Jin Han. Derek, so maybe can you some comment to the audience?
Yeah, hi, good afternoon everyone. Welcome to this Basic Research 1 session. I'm Derek House-Roy from Duke US in Singapore. I look forward to hear the talks in the session. Thank you.
Jin?
Same. Good afternoon.
So actually the first speaker will be introduced by the Jin Han, please.
Yes. First speaker of this Basic Research 1 speaker is William Boisbert, who came from Hawaii University, who will give a talk about regulation of crosstalk between perivascular adipose tissue and atheroma. So please, William.
Thank you. Thank you for the introduction. I have a bit of a cold, so if my voice gets a little jammed up, you'll know why. So thank you for the introduction, and thank you also for the invitation. As always, it's a pleasure to come to this meeting. And I'd like to especially thank Dr. Kim, although he's not here, Kim Moo-hyun, for always inviting at least Eric and me to this session every year. So what I'd like to talk about today is a little bit different topic than I've been presenting over the years, looking at the role of perivascular adipose tissue and atheroma development, and then introducing a therapeutic compound called compound 21 in the process. So in the way of introduction, I'd like to talk first about the renin-angiotensin system in vascular function. I'm sure you all know the renin-angiotensin system, or I'll call RAS. So RAS, as you know, plays a key role in various vascular function, especially in blood pressure monitoring. And angiotensin II, which is the main mediator of the RAS, interacts with angiotensin II type 1 and type 2 receptors, which is really the focus of the talk today, which are both G protein coupled receptors with multitude of signaling that's downstream of the G proteins. AT1R is expressed ubiquitously, whereas AT2R is expressed highly in fetuses, but not so much in the adults, except in certain pathogenic cases. So AT2R expression is associated with anti-atherogenic properties, just to cut to the chase, such as increased vasodilation and reduced inflammation and fibrosis. And compound 21, as I mentioned, is a known agonist of AT2R among many agonists that we know about already, which induces vasodilation mainly by releasing nitric oxide. And then the other introduction has to do with perivascular adipose tissue. This is not a very well-known organ, if you will, that has been known to modulate atherosclerosis, but there is some information out there relating to the role that the PVAD plays in atherosclerosis. So it's thought to regulate the development of atheroma, and the location of atheroma formation can depend on the surrounding PVAD, on the adventitia of whatever vessel that you find it on. And PVAD-mediated regulation of atherosclerosis may be affected by the type of adipose tissue that's either white, brown, or beige surrounding the vessel, but very little is known about the molecular details of how PVAD can actually influence atherosclerosis. And C21 has been shown, the compound 21, to transform by our group with collaboration of a Spanish group, that I've been working with, to transform white adipose tissue into brown or beige adipose tissue. So here, this is a cartoon that kind of summarizes in a very simplistic way, what is known, what the paradigm of how PVAT can regulate atherosclerosis. And on the left here, anyway, on the left here, it shows the healthy homeostatic vessel wall and uninflamed or normal fatty acid or adipose tissue, which can secrete compounds like adiponectin, nitric oxide, and IL-10 that stabilizes the vascular function. However, when it gets inflamed, as you see on the right, it can secrete products like leptin, visfatin, fatty acid binding protein, as well as cytokines such as MCP1, TNF-alpha, and IL-6 that can draw immune cells into the adipose tissue, and then also contribute to the atherosclerosis that you see on the bottom. So I'll do sort of a reverse of how we did this study. I'll go through the in vivo study first, just to give you the main result. So we first planned to do an atherosclerosis study using LDL receptor knockout mice on both chow as well as in high-fat diet, and then giving either a vehicle, which is the C, or C21. C21 was given by gavage on a daily basis. This was a lot of work for the students and the postdocs who were involved in this. So this went on for 12 weeks, and then we did all the measurements that you see on the right, the sample, the plasma, as well as the thoracic and abdominal aorta sections, and then the fatty acid or the adipose tissue surrounding the thoracic or the abdominal PVAT, as well as a heart function. So the aortic tissue analysis that was done in A in the green, you see we did the en face oil red O staining, and then B is where the thoracic aorta section was isolated from, and then C is where the abdominal aorta section was isolated from, and those were all will be shown below. So this just shows that the PVAT surrounding the thoracic and the abdominal aorta. In A, you see thoracic PVAT, and then in B, you see the abdominal PVAT. And you can probably notice that the density with the H&E staining around the vessel is quite dark in the thoracic PVAT, and it's much lighter white adipose tissue that you see around the abdominal PVAT. So when we do the atherosclerosis staining, this is more like a preliminary study because we just began this study. We have a lot more animals going, but just to show you what happened here with the few animals that we already did, we could see that the plaque area covering the inside of these aorta was significantly reduced in the C21 treated animals. And then when you look at the sections that I mentioned, the thoracic PVAT and abdominal PVAT area, you can also see with the chow diet on the left, there was no atherosclerosis to speak of. And with the high-fat diet, you can see atheroma forming in both the thoracic and the abdominal areas. And then with C21 compound treatment, this was much reduced, much like the chow diet. As I said, we have many more animals to do to get the numbers up so that we can do some proper statistics on this. So this slideshow, I'll do the B first. B is the atherosclerosis that was induced by 4 weeks, 8 weeks, or 12 weeks of high-fat diet. And you can see that AT2R is not really expressed at 0 and 4 weeks, but it starts to appear at 8 and 12 weeks. So with 12 weeks especially, we can see that the AT2R is robustly expressed. And then on the left, in A, you can see both the AT1R and AT2R expressed in control as well as C21 treated animals. This is after 12 weeks of diet. And especially the AT2R with C21 was robustly expressed in these atheromas. So then we looked into some of the mechanisms of how C21 might affect the vascular function. So we measured The nitric oxide contribution, which is you see in A, and you can see with high-fat diet in the third bar. Does this work? Oh, yeah. You can see that the nitric oxide has decreased with the high-fat diet, but then with C21 feeding, you can see that it went up to the levels of the chow-fed animals. And then we also did some collagen analysis that you see in B. So with the high-fat diet, again, you see a high increase in collagen deposition in the vascular wall that you see in the bar here. But then with C21 treatment, this was back down to about the level of the chow-fed animals. We also looked at matrix metalloproteinases like MMP9 and MMP2, which were raised in the high-fat diet-fed animals, but then again came back down with the C21 treatment. This is looking at the stiffness or contractility, if you will, of the vessel in the abdominal aorta section. And you can see with a high-fat diet, the controls are down here, the high-fat diet, there's an increase, significant increase in the contraction of these little segments of the aorta, as well as relaxation. But then with the C21 treatment, as you see here in the triangle, it went back down to the level of the chow-fed animals. We also wanted to know if there was, as I mentioned earlier, C21 has known to have an effect on the white adipose tissue, transforming it into more like a beige type of adipose tissue. So we wanted to know if that indeed was the case with C21 treatment. And so with these human endothelial cells, we treated them with C21 and also with AT2R antagonist, the PD compound, as well as losartan. So you can see with the C21 treatment in the red, you see the increase in UCP1, CDA, as well as PPAR gamma, which are all indicative of more like a brown adipose tissue signature. And some of these AT2R antagonists were able to bring it down a little bit, especially with the PD compound, but not so much with the losartan. And here we're looking at oxygen consumption rate. So in the black, you have the control animals, and then in the red, we have the C21 treated animals. And you can see that the metabolic rate is much higher. The oxygen consumption rate is much higher in the C21 treated cells. And then you can see other properties here. So this is all indicative of the white adipose tissue turning more into the beige phenotype. We've known from the past with our previous research that the AT2R can also bind to what's called MOS receptor as well as bradykinin receptor. And so we wanted to know if that was the case here with using human endothelial cells. And you can see with the co-IP studies that there are indeed expressed together, or at least they bind together. And here with the C21 treatment, we can see that the MOS receptor co-IPing with the AT2 receptor went up significantly, as well as MOS receptor co-expressing with bradykinin receptor went up significantly as well. And with these cell staining, you can see co-staining of MOS receptor and AT2R, as well as bradykinin receptor and AT2R here as well. And then just give you one slide on the signaling mechanism. We knew from our old data that PKA and AKT are involved in the increase of nitric oxide that's mediated by C21. So we wanted to confirm this, and we can see by staining for the release of nitric oxide, C21 definitely has a big increase shows a big increase in nitric oxide. However, when you treat them with KT 5720, which is a PKA inhibitor, as well as tricerbine, which is a AKT inhibitor, you can see much less release of nitric oxide by these inhibitors, and then there's almost none. by, the, by adding those two together. So what it comes down to is this kind of a final slide. What we think is going on is we think that the bradykinin receptor, MOS receptor, and AT2 receptor are actually trimerizing. We still need to prove this. And as they trimerize, they go through pKa and AKT to increase eNOS production, which I didn't show you, but it does happen, which increases nitric oxide, and that results in the vascular protection. So that's the left side here. And then AT2R alone, this is what we've also found through this study, results in the C21 acting through AT2R results in decreasing the MMPs, the 9 and 2 especially, decreasing collagen as well as TGF-beta, which also decreases or relaxes the artery much more. So what we're trying to do right now is to analyze the PVAD itself. You can see here, get brown adipose tissue and white adipose tissue down here. So we're trying to see if P21, C21 treatment actually has an effect on the phenotype of these adipose tissue around the aorta. And we also want to know if there's a sort of a crosstalk, as the title suggested, between the adipose tissue and the aorta by looking at extracellular vesicles that we'll be isolating from these animals that were fed a high-fat diet. So I'd just like to acknowledge Jason Ire, who is right here, and Marta Viana, who is the representative from San Pablo University in Madrid, who is right here. She did a sabbatical in my lab for a year. So they're the most responsible for this project, but many other people are involved in the project as well. So thank you very much, and we'll get some questions later on.
Okay, thank you, William, for keeping exact time. So we will have discussion time after all speakers complete. So Derek, please.
Okay, great. It's my pleasure to introduce the next speaker, who's Professor Hyo-Sung Kim from Seoul National University Hospital, who will talk on development of biotherapeutics for myocardial regeneration using latrofilin-2, a marker of cardiac progenitors. Thank you, Professor Kim.
Thank you. For the next 50 minutes, I'd like to touch on about 10 years research on latrofilin-2 in our laboratory. There are several strategies of therapeutics for heart repair. First is cell-based therapy. We can implant cardiac progenitor or cardiomyocyte directly, or we can achieve the transdifferentiation in situ by gene transfer to the heart. For this therapeutic strategy, we try to identify a new surface marker of cardiac progenitor. That was latrofilin-2 that we found for the first time 5 years ago. That is novel surface marker for cardiac progenitors as well as mature cardiomyocytes. 5 years ago, we first report identification of latrofilin-2 in circulation, and then we identified signaling mechanism of latrofilin-2, and we achieved myocardial repair by transplantation of LATRO2 positive cardiac progenitors. The first paper is shown here. We established protocol of differentiation of iPS cells toward cardiomyocytes. And during this process, we employed suspension curch to make cardiosphere under this curch condition, then changed attachment condition to make beating cardiomyocytes. About 10 years ago, this is about 10 years ago, at that time, the marker of cardiac progenitor were double positive for PDGF receptor alpha and FLIC1. That is suggested by Gordon Keller. Thus, in day 0, about 5%. And day 4, with different conditions, we can enrich progenitor up to 11, 30, or 90%. Thus, using this 4 population, we try to find the new cardiac progenitor marker. We guess marker may increase its expression while the enrichment process from population 1 to 4, then we found about 10 genes that continues to increase the expression while the population from population 1 to 4, among them only one, latrofilin 2, is surface marker as well as it is functional marker because it is G protein coupled receptor. Then we established knockout mice. Hetero knockout mice were viable, but homo knockout mice died in utero because of malformation of heart. Then we found the signaling mechanism of latrofilin 2, phosphosarc, phospho-CDK5, P38 MAP kinase are key proteins that are activated and phosphorylated to convey cardiac lineage commitment of stem cells. Furthermore, we found really high homology between mouse and human. The data that we obtained from mouse can easily applicable to human. Then we achieved myocardial repair after infarction by transplanting lateropositive cells. Lateronegative cells cannot achieve such kind of repair. Histologically, here is condition, PBS conditions. The white is resident cardiomyocytes. They express also latrofilin 2. And we transplant green lateropositive cells. Green cells are enriched here, and they express latrofilin 2, and then they differentiate into cardiac cardiomyocytes expressing white sarcomeric actin. When we transplant green lateronegative cells, they did not differentiate into the cardiomyocytes. Then we tried to guess latro2 may play a crucial role in the regeneration and repair process of cardiomyocytes in adult heart because it is functional marker and it is regulator of differentiation that is similar to regeneration, and it is not only expressed in progenitor but also in cardiomyocytes. Thus, we try to make cardiomyocyte-specific tamoxifen-inducible latro2 knockout mice it took about 3 or 4 years. We started latro2 flux mice and alpha mice having chain promoter-driven MER-CRE-MER. And after crossing these mice, we can achieve such a hetero and achieve the homo. Thus, if the cells are cardiomyocytes must have chain promoter is activated and CRE enzyme may be produced independent of tamoxifen. Thus, it is activated, it attacks LOXP site and delete exon 3 of latrofilin 2, generating tamoxifen-inducible cardiomyocyte-specific latro2 knockout mice. When we introduce tamoxifen, it binds MER and activates CRE only in cardiomyocytes, leading to deletion of exon 3 of latrofilin 2. When we administered tamoxifen about 4 days, latro knocked down, protein disappeared, and remarkable dilatation of heart. Really remarkable. Histologically, this is normal mice, knockout mice. Latrofilin is totally shut down. Myofibers are separated. VT, high-degree AV block happened. LV dilated, contractility decreased, and poor survival. Under situation of AMI, the prognosis is even worse. Fibrous area increased. Here, scar is really increased when latro2 is knocked down. Remarkable. High degree arrhythmia happened, really poor survival. Thus, we tried to improve a little bit survival because of the number of the animal that can be analyzed. Thus, we reduced those down to 20 milligrams per kg per day. For 7 days, latro is sufficiently knocked down, protein disappeared, and heart dilated. Survival is a little bit improved. Thus, we can get sufficient number of mice dilated even with a small dose of tamoxifen, thin wall, dilated wall. Here, histologically, there's been well-demarked Z-disc in the control conditions. Tamoxifen 20, 70, that's mean this disappeared and fibers separated. Z-disc disappeared. A TEM electron microscope shows a remarkable finding. This is control conditions, well-aligned myofibrils with Z-disc. Between the space, there are healthy mitochondria. But if latro knocked down, there is a really dehiscence of myofibers and destruction of mitochondria. Here, dehiscent myofibers and destroyed mitochondria below the Z-disc. Thus, we focus on two main phenotypes. Myofibers separated, mitochondria destroyed. Here, mitochondria is seriously damaged when LATRO2 is knocked out. To find out the mechanism of mitochondriopathy in LATRO2 knockout mice, we did a transcriptome analysis between wild and LATRO2 hormone knockout embryo and focused on mitochondrial organization gene clusters and found PIPA gamma coactivate 1 alpha is really shut down with latrofilin 2 knockout. This is post-regulator of mitochondria. Thus, we found latro knockdown, PH1 alpha is also knocked down here. Only heart, not brain. Histologically, here, PH1 alpha and nuclear transcription factor disappeared when latro knocked down. That leads to mitochondrial destruction. We also analyze mitochondrial function, mitochondrial membrane potential using isolated single cardiomyocytes from LATRO2 knockout mice and put JC1 dye. If it turns red, membrane potential is good, not, it's bad. Thus, JC1 tagged with FITC applied in vehicle, red color appeared, but latro is knocked down, red color is really decreased. In control conditions, about 30% of mitochondria isolated was a red color, healthy. But latro knockout, the number percentage is less than 10%, about 4 or 5%. Result is accumulation of ROS. Mitochondria destroyed, then ROS generated. Here's ROS non-4-hydroxynonenal marker is enriched when latro is knocked down because of mitochondria destroyed. Another point is decent myofibers, like this. Thus, we also focus on the cell-to-cell junction molecule, adhesion molecules in this comparative transcriptome analysis and found there is a huge reduction of gene clusters involved in cell adhesion and cell-to-cell junction in LATRO2 knockout embryo. Among them, we focus on cadherin 2. That is consistently decreased in cell adhesion molecule cluster, straight muscle cell differentiation gene cluster, and beta-catenin binding gene clusters. In addition to cadherin 2, there are several genes depressed in LATRO2 knockout mice that are involved in cell-to-cell junctions. Here is histology. Normal condition, we can see clearly NCAT that is colocalized with junction. LATRO knockout disappeared. Connexin enriched constitute gap junction disappeared with cell separation. Desmocolin 2 disappeared. Thus, most of the cell junction protein disappeared after LATRO knockout mice, and then we also analyzed the situation after myocardial infarction. Peri-infarct myocardium at 2 weeks after MI, we can see clearly NCAT disappeared. Connexin disappeared. Desmocolin disappeared. To find out the regulator mechanism of why such kind of cell junction proteins are decreased, we remember CDK5, SRC, P38, MAP kinase are key downstream molecules of latrofilin 2, like this. Latro knockout mouse shows decreased phospho-CDK5, SRC, and P38. Then we focus on these downstream molecules and found MAP kinase cascade are systematically shut down by lateral knockout, followed by decrease of junctional proteins such as desmocolin, desmoglein, and connexin. Here's protein data. Latro is knocked down with tamoxifen. Latro 2 is knocked down. P38 is shut down with junctional proteins. Thus, we guess when we use P30 MAP kinase activator, such as anisomycin, we may recover such kind of phenotype, mitochondriopathy and junctional proteins. Thus, we put anisomycin, P38 is activated, PGH1 alpha is activated, and junctional protein restored. Phenotype is also partly recovered. When we treat tamoxifen and made kind of dilated cardiomyopathy, and when we treat these animals with anisomycin, we can reduce the dilation like this. In TM, this normal, tamoxifen, anisomycin fairly recover desmosome, adherens junctions, and Z-disc are all recovered. Here, desmosome, adherens junction, desmosome, adherens junction, gap junction is like this, and mitochondria is good, destroyed all the structures, and anisomycin recovered partly desmosome, adherens junctions, mitochondria, and gap junctions like this. Protein is recovered, depressed, recovered by P38 activators. Histologically, PGH1 alpha is rescued. Then mitochondria is rescued. ROS disappeared. Junctional proteins are rescued. Desmocolin rescued. Connexin rescued. We finally try to find association of latrofilin gene mutant and human heart disease using Genome Ed or UK Biobank. In this bank, we found the ratio of observed patient versus expected patient is very low, suggesting latro variants may be fatal in human. And UK Biobank, we tried to find latro mutants and 9 phenotype of heart disease, only 1. Such a cluster may be associated latro variant. That disease cluster is other congenital heart disease. To summarize my story, when it is normal, latro2 activate P38, leading to PGH1 alpha activation and healthy mitochondria. Concurrently, it may induce normal expression of NCAD, desmocolin, desmoglein, and connexin. That is important constituent for adherens junction, desmosome, and gap junction. If it is knocked down, all signals shut down, leading to dehiscence of the muscle fiber and accumulation of ROS, leading to DCMP. When we supplied P38 activator, we can partly rescue mitochondria as well as junctional protein. Population and circulation research, and we add one more step to our journey. Now we are trying to apply latrofilin 2 gene therapy or developing latrofilin 2 agonistic antibody to regenerate heart. Thank you very much for your attention.
Great. Thank you very much, Professor Kim, for that excellent talk. Now my pleasure to introduce the next speaker, who is Professor Hsien-Woo Cheng from Yanbai University, who will talk about the impact of chronic stress on adipose and vessels, focusing on the PAI-1 and DPP. Thank you, Professor Cheng.
Thank you, for Chairman. Also, thanks to the Professor Kim Moon give me a chance to present again in Busan. So as known as psychotic chronic stress is an interactive risk factor for the CVD, as known as many risk factors for the CVD. For example, in aging, smoking, and diabetes and hypertension. So kind of the stress can be controlled by the drug and some method. But psychotic stress is not so easy to control in the disease. So this is the first report in 30 years ago. The animal studies the acute mental stress influence the insulin sensitivity and plasma neurohormone, stress hormone. And so here is that in earlier time is that finding the stress hormone includes the, for example, adrenaline and corticoid and growth hormone, so on. So early time responds very quickly, but insulin level is still includes the long time. Another clinical paper showed that the moderate stress and influence the AM patient. And so here, very easy to look after the stress, where higher includes the AMI incidence. There also one paper published 6 years ago in the level of leptin and adiponectin, because the adiponectin is very good molecule in from the adipose tissue. So I was interested in adiponectin and related factor. So here is so that the leptin is very included after stress in between the resistance, insulin resistance, and sensitivity. And finally, then the adiponectin is quite different pattern is decreased. And also earlier, we also one basic research published one diabetes finding the stress accumulated macrophage accumulation in the subcutaneous adipose tissue. It also find the insulin resistance and impaired happened. So one is a very interesting paper 10 years ago in the Nature Medicine is published chronic stress compared in the medical doctor in the off-duty and on-duty ICU doctor, finding the stress score includes the on ICU. Also, peripheral blood leukocytes were higher in this kind of doctor. So another paper published in Nature, sleep modulation hematopoietic stem cell in protected against ulcers. Here is finding the stress impaired and hypercollagen level is hypercollagen regulated hematopoietic stem cell proliferation. So because of after stress and then reduction in the hypercollagen and stimulated hematopoietic stem cell activation in the many monocyte leukocyte released to the peripheral blood accumulated in the atherosclerosis plaque. And we also one interesting paper published in Nature from the Harvard University. It is very simplified study, but it is published in Nature. It is complex stress, it is capsaicin and resistant stress affect the hyperactive sympathetic nervous drive the depression of the melanoma stem cell. Melanoma stem cell is in the activated and grows out in the move to the another place. So the hair is just one way stress in the hair is changed the gray. So it is kind of study is accumulated while do the stress work. So this means time limit. So why do we focus DB4? As you know, DB4 we know is the diabetes field. So D4 is many effect, but D4 is very higher degraded GLP-1. So DB4 and degraded GLP-1 and affect the glucose level. So I just screened it in the many tissue after 2 weeks stress, find the vessel and the small intensity lung and kidney, so on the many tissue. I find that after stress, includes the DB4 level, but unfortunately cannot detect the bone marrow in the D4 level 0, no action. So I did the chronic stress in the focus and DB4 and GLP-1 and in the several study. First study in the just 1 month stress and analyze the several work. So after stress, the body weight reduction and the circulating DB4 level is quite changed. And so here, and almost 10 times in the after stress, not only the brain and we also find the blood is very increased, time dependent increased. It means maybe D4 is very activated in the after stress and the GLP-1 pathway worked in the body. So we are just studying the HE staining and the DB4 staining. I find that the D4 also included in the brain. So I just Fox analyzed the hematopoietic stem cell. I just hematopoietic stem cell identified as a 4 marker. First in CD1 positive cell, and then we also did the CD48 minus CD140 is a positive cell is a hematopoietic stem cell. After 2 week stress, I find it not only in the so here in the bone marrow is a very higher number. Also the cell cycling also analyzed included after stress. Also some staining here. So here the CD140 50 the positive cell is after stress in the bone marrow is a high number. So for mechanism study we just did in the bone marrow and isolate the positive cell, analyze the GLP-1 receptor. And the other receptor 3, I find the other receptor were higher, but the GLP-1 receptor is decreased. So in the bone marrow, and so I just used the DB4 inhibitor, checked the effect. Find the time dose dependently reduce the hematopoietic stem cell in the peripheral blood. And so here the identified in the CD150 the marker positive cell reduced in the dose-dependent effect. So, well, here is where in the peripheral blood also we analyzed here LY6C higher level monocyte. And the neutrophil also includes the reduction in the by DP4, but this year no yet dose-dependent effect. And the bone marrow, I will just use the 4 mechanism. We also did exenatide. And so here, exenatide after treatment, we find the hematopoietic stem cell in the bone marrow with reduction. And then this can also study the bone marrow with the GLP-1 receptor and find it include after treatment. So, and we just use the DB4 knockout mice. GLP-1 knockout mice, and find the bone marrow is a GLP-1 receptor is includes the, sorry, this is not mice. So I find the includes them. So I conclude this first part of the story. Chronic stress in the DB4 level includes the degradation, GLP-1 degradation very higher, so it reduces the CXCR4, but today is not so the CXCR4 data. But yeah, another way is the other step 3 is included. But we just use the DB4 knockout, DB4 inhibitor or GLP agonist, to reverse this kind of change and reduce the hematopoietic stem cell activation and so on, and improve the peripheral blood in the neutrophil and the monocyte. So second topic is about in the atherosclerosis androgenesis. We just used in the upper knockout mice. So here is still the upper knockout that check the blood DB4 GLP-1 advanexin. To find it in the even in the upper knockout the hyperdiet stress. Find the combined stress by the D4 level includes the GLP-1 decrease and the same part of absorbed apodipronectin. So here is a way just use the exenatide. Exenatide is improve the macrophage infiltration and the inflammation cytokine expression and so on. And we also the cytology analysis regarding always staining and some functional staining so on. I find exenatide improve the vessel matrix metabolism. So for mechanism study, we just treat exenatide pre-adipocyte. I find the exenatide stimulated adiponectin MR expression dose-dependent effect. So, and we also analyze the brother, the leptin is included and adiponectin is after stress is included. Sorry. Oh, no, sorry, second mistake. Adiponectin is reduced. And furthermore, and the next change to the in the angiogenesis, we just stress model, use the angiogenesis model. I find that DB4 knockout mice. I also find the angiogenesis is preserved in the stress situation. And for also studied in the capillary density as improved the DB4 inhibition. We just use the DB4 dose-dependent effect. And we are further confirmed, we also still use the GLP agonist. We find the agonist myriad ischemic induced angiogenesis. So next, finally, we use the adiponectin knockout mice. Even we just use the exenatide, but did not any effect observed in the adiponectin knockout mice. So it means stress is still working the DB4 GLP-1 pathway, and adiponectin one way is in the prevent atherosclerosis formation, another way is stimulated angiogenesis. So next, we just study the stress model due the D4 the deletion prevent adipose differentiation. For mechanism study, we just use the analysis for target several molecules about the adipose differentiation and the adipogenesis related molecule. We find the D4 knockout mice. We find some, of course, I just most of the time studied cardiovascular here. However, yes, this kind of the D4 level is encouraged that the adiponectin and PPAR gamma, so GLP-1, so on. So easy hero studied after stress two type genetic mice. In the stress, wild type mice, the adipose cited the differentiation, but DP knockout, the adipose site includes, it also find that in the inflammatory infiltration is blocked. So serum level, we just use the stress serum, and here is find we just use the cardiovascular silencing and checked in this way. I find that after stress serum transformation is differentiation were promoted, but in the cardiovascular silencing, where they prevented this change. So I summarize this result is chronic stress still is DB4 GLP-1 pathway. This is focused in adiponectin. Adiponectin is through the cathepsin, as and through the PPAR gamma and one molecule, and prevented the differentiation and the worked well. And the last part is some few slides so the CD8. Here is all study I just studied focusing stress. Yeah, so here is the stress. We are higher stimulated aneurysm formation. But for mechanism study, we are NLRP3, we are included in the, but in the knockout, CD8 knockout mice somewhat prevent this kind of change. So LHDRO study is stress area promote vascular matrix protein metabolism and macrophage infiltration. But CD8 knockout mice reduce the macrophage infiltration and prevented this kind of matrix protein degradation. So, and this last one study is in the ischemic model, where ischemic model find the ischemic can higher activated CD8 in the, and so here, knockout, of course, no change, no detect, but the ischemic includes the peripheral blood CD8 cell and the ischemic muscle, the CD8 cell is accumulated. For mechanism, we just with the CD48 knockout mice. were 2 hours surprise. See the CD8 the deficient mice as promote stress-related angiogenesis. And so here, and for mechanism study, we just still work in the NRP3 and the VGFs in the knockout mice level were included, and this NRP3 reduced. And for mechanism study, we also in interferon gamma knockout mice where yield the same conclusion with CD8 knockout, CD8 deletion mice. So adaptive transfer with CD8 minus T cell yield the same conclusion with CD8. It means with the adaptation transportation, CD8 deletion cell find the here, so the recover, they even used to the CD8 wild-type mice. They are recovered by the adaptive transportation. So last thing, the PI1 is still worked in the stress. PI1 knockout is 2 hours surprise. PI1 knockout promote, accelerate the C8-induced AA formation. And so mechanism study, so here. So still I will back to the Yanbian. I still all work in the, for example, is thrombosis, also muscle, and so on. So we enjoyed the stress work. So this just in, I worked in Nagano University 20 years. I just sitting here, and now it's changed to the Yanbian. Our department, so many students, also their staff. So in Japan, it's many stress, but you enjoy the Japan life. So that's in the Hanami, I just drinking too much. So thank you so much.
Great, thank you very much, Professor Cheng, for that very good talk. Thank you.
So I'd like to introduce in the last speaker, the Hyung-Kyu Kim from Inje University. He will talk about the multifaceted role of cerebron in heart failure from metabolism to ion channel regulation. Professor Kim, please.
Yeah, thank you very much, Young-Gun, the chairman. And then it's my great honor to have the chance to present my study in the JShare 2024. There's no COI. And as you know, everybody knows that cardiovascular disease is the world top cause of death, not only in the world, even in the Korea, during the last 20 years, the prevalence of the heart failure, hospitalization, incidence, and mortality is rapidly increased in Korea. And as the medical drugs, the major medicines are generally used are RAS blockers, beta blockers, the diuretics, and statins. And recently, the SGLT2 inhibitor is a catch up the market and increase that the using of the SGLT2 inhibitor. But still, we need something more to taking care of these heart failures. So there is generally 2 types of heart failure cause. The first one is the infarction or ischemia, the myocardial infarction starting and expansion of the impart. Generally, they go to the global remodeling. Or another one is under some metabolic stress. So we can call it also some metabolic cardiomyopathy. So that is under the hypertension or diabetes and metabolic stress enhance the hypertrophic heart, and they are going to the dilated heart. Even that these are different kinds of the cause, but finally they are final goal or final that end phenotype of the heart failure is a loss of their contractility, and that is the major outcome. You know, maybe it is a very basic of the cardiac physiology. The excitation and contraction coupling is very important in the heart. So initiation of the calcium entry through the anti-calcium channels, and that mediate the contraction and reuptake to the SL or removal through the NCX. So the tight regulation is very important for heart contraction, and this imbalance can cause the heart failure or loss of the contractions. So there's several studies suggest the anti-calcium channels density and regulations are altered in the failing human ventricular myocyte. But compared to their protein level decreased density, but the anti-calcium channel coding genes are comparable between the control group and heart failure groups. So what is the mechanism of the anti-calcium channel reduction in the heart failure patient is this questionable. Move to the cerebrons. So cereblon is a substrate recognition component of DCX E3 protein ligase complex. So that is not working alone, but he is or she is a very good role, important role to catch their target protein and degradations. This is their 2D structures. It's composed by the 441 amino acid, and there's a two important domains, the non-substrate binding domain and drug binding domain. They are actually very close. They are working together, and they have some interaction between two domains. So cereblon is introduced 2004 in the Joseph Higgins. He is finding the mutation of cereblon in the mild mental retardation families. And the next year, Charles Park, who is our collaborator, finding out the role of the cereblon in the brain as a binding protein for the large conductance calcium-activated potassium channel in the brain. So maybe the kinds of the ion channel functions can maybe regulate some mental retardation in the human families. After 5 years, there's a very important finding was reported by Hiroshi Honda. He found that cereblon is the major binding target of thalidomide. So maybe you know these kinds of the features, the children without their limbs. So thalidomide is an immunomodulator drug. It's developed 1957 in German. So they are intended for the morning sickness. So they are very working well for the pregnant women, but after 10 months, when they gave the children, they showed the teratogenicity of malformation of the limbs. And worldwide, 12,000 infants are born with pocomelalia and 50% of bone deaths. That is known as the worst drug accidents. But they don't know why these kinds of drugs working in that way, bad way. And he find cereblon binding the thalidomide. And this is the mode of action of the teratogenicity. So cereblon should working during the development. It should degrade some proteins important for the development, but this IMID is inhibit these kinds of binding, and the substrate accumulation can cause these kinds of the teratogenicity. And after 3 years, there's another big finding was suggested by the Choxenbach again, and he found very important role of the cereblon as a regulator of the energy metabolism. CRBN knockout shows the highly increased phosphorylation of AMPK alpha. As you know, AMPK is a major regulator of the energy homeostasis. And this whole body knockout mouse show the anti-obesity properties against the high-fat diet. And even this CRBN deficiency prevent fatty liver in the high-fat diet mouse model. After several years, he found a very clear mechanism of how this cereblon enhance or cereblon targeting the AMPK, because cereblon can degrade AMPK gamma subunit. This gamma subunit is important for the phosphorylation of the AMPK alpha subunit. So decrease of cereblon is more AMPK gamma subunit, and that enhance the phosphorylation of AMPK alpha. And the next year, the science and nature found out not only that kinds of role, CRBN also working as the anti-cancer effects. So this kinds of mode is something like that. EGFR1 or 3 is a very important protein for the cancer biology, but they are not actually target of the cereblon, but in the presence of IMID, they enhance their affinity to this important protein, and they degrade this more to making the anti-cancer effect. So until now, about 20 or 30 different types of the target of cereblon was known, and depend on the tissues or target, they can regulate the different types of role. So we are focusing on the heart and mitochondrial study. We are checking what is role in the heart. So first, we start with the NCBI, the database. Dilated cardiomyopathy patient actually showed a higher CRBN level. And not only the database, when you collaborate with the Ipesimis, he found out the low ejection fraction patient, their heart mRNA showed a higher CRBN level in their heart. So what we think is a cereblon can regulate the energy metabolism and also can regulate the electrical stimulation through the ion channels. So it can be maybe master regulator of the heart. We get the CRBN knockout mouse and checking their heart functions. they showed a little bit hypertrophy and little less the weight gain. When you do the echocardiograph, they show the significantly enhanced contractility in their heart. And not only the living heart, when we take the heart out of the body, ex vivo study, there the isolate heart still show the higher contractions under the PV roof systems. So not only heart system, but when we isolate single cardiomyocyte, left ventricle, and then when we stimulate with the electrical stimulations, that the cardiomyocytes still have these kinds of the hypercontraction activity in the CRB knockout mouse. So we checking first the energy metabolism aspect of this mouse. So when you do the metabolic cage study, 2 days they show the higher energy expenditure in this model, and their cardiac mitochondria has a more dense and more higher oxygen consumption rate, and making the more ATP than the wild type. And what we think is the cause of these kinds of phenotype is enhanced AMPK alpha phosphorylation. Then we move to the calcium study. And in the isolated cardiomyocyte, we're checking the calcium transient. The calcium transient is higher in the knockout mouse than the wild type. And that these kinds of the enhanced calcium signal enhance the action potential duration in the ventricular cardiomyocyte. And when you're checking the L-type calcium channel by electrophysiology, they show the higher current, the density, and the activity in the knockout mouse. Is that the effect of the transcription or post-translational regulations? When you check the mRNA level of the CAV1.2 protein, there's no difference between the gene mRNA level, but the protein level only enhanced. So we are very cleared. This one is not the transcriptional, but post-transcriptional regulations. We check the CAV1.2, if that is localized via CRBN, there are good localizations. And we do the in silico simulation if they can have the possibility of binding up to these two proteins. And we go to the real studies, the biochemical studies. CAV1.2 and terminal can be bind to the cerebellum, the lone domain and terminal. And not only the interaction and binding, they actually enhance the ubiquitination of the CAV1.2. And very interestingly, the thalidomide, these drugs can interrupt these kinds of the binding between CAV1.2 to the CRBN. And that it's luckily we found we collaborate with our Korean chemical institute. They found a special product TD165. They can very effectively remove or degrade CRBN itself within very short time. So when you treat it with a pharmacological inhibitor, TD165, there is a CAV1.2 is increased in the two types of the cell lines, HEC cell and AC16 cells. So in the mouse model, when you test it with the doxorubicin-induced heart failure model, the CRBN knockout has a much higher contractility. And also, the myocardial infarction model, they show the higher contractility compared to the wild type. And when you do the overexpression of cerebellum, these kinds of increased contractility is attenuated at the normal level. This kinds of study is also reproducible in the CRBN cardiac specific knockout mouse that show the higher contractility in this model. So this is our summary. CRBN is highly increased in the heart failure model, a patient, and they can cause the CRBN, the CAV1.2 degradation and decrease the contractility. There's one shot more studies. Not only the CAV1.2, we found some target of cerebellum, and there is a TRPC channel. So TRPC channel is important for the structural the modulation or regulation of the heart, especially they are known as a link with cardiac hypertrophy. As I showed you before, we found that there's a small hypertrophy in the wild type knockout mouse, but they don't have any AMP or maybe pathological hypertrophic marker or fibrosis. So it's something like the more physiological hypertrophy. And in the isolated cardiomyocyte, even in the basal level, calcium level is higher. And this is the very key point of our studies. When you do the verapamil, anti-calcium channel blocker, their contractility is decreased than the wild type, but hypertrophic, the phenotype is still be there. So anti-calcium channel increase is not the cause of the hypertrophy. So that's why we found out another target, and we found out that TRPC1 is highly increased with the CRBN knockout mouse model. And when you do the knockout in the in vitro system, that can show the same result. When we express the TRPC1 in the hex cell, the CRBN knockout, much higher expression of the TRPC1. And in these kinds of the knockout cell, when you treat it with the calcium entries and then checking the basal calcium changes, this one is without that voltage or electrical stimulation. Still, the calcium can be enhanced in the CRBN knockout and the TRPC1 overexpression cell model. We do the directly checking the electrophysiology study with the TRPC1 channel, and this CRBN knockout enhance the TRPC1 activities directly. And same the method we use, and then we found that the N-terminal of the TRPC1 is binding to the CRBN. As finally, we found that the TRPC1 is another target of cerebellum in the heart, and that can regulate the physiological hypertrophy. So as a take-home message, CRBN can degrade the AMPK or anti-calcium channel and TRPC1, so they can modulate the metabolic regulation and contractile regulation and then structural regulations. Thank you very much.
Thank you very much, Professor Kim. 불 좀 켜주시겠어요? Turn on the light, some brighter. So actually now the discussion time is approximately 20 minutes can be for discussion. So the panelist is from the Soo-jin Kim, and the Lang Hong, and the Jiang-woo Chen, and the Young-suk Kim. So actually, because we have some sufficient time, at first we have a discussion from the panelists. So who is to start to make a discussion? The Young-suk, please.
Thank you for the wonderful and grateful lectures from the four speakers. First, I have some questions to Dr. Hyung-kyu Kim. So, yes, as you mentioned, energy metabolism might be changed with age. So I'm wondering the expression of CRBN, the expression level of CRBN would be different in the aging-related disease or just a biological aging status?
Yeah, we didn't checking in our model, but when we checking the database, there is several good database with agings. We found that cerebellum level is increased in some specific tissues with human agings. So we didn't check very carefully, but we found that the aging may be enhanced the CRBN level, and that can cause some kind of over degradation of the key proteins. We can maybe enhance the age-related disease.
Okay, on one more question. And in the earlier slide, you showed the mRNA levels of CRBN was not different, but the CRBN protein level was quite different. So I'm just wondering, the PRBN has the protein of CRBN has the modified by PTM to change their protein stability or their half time?
The data, what I showed is the cereblon level. That is the CAB 1.2, the mRNA level is not changed, but CAB 1.2 protein level is changed. So that's why we think that is a direct binding and degradation. And actually that is samely we found with the TRPC channel. So the mRNA level is not changed, but only the protein level is changed. So I thought these post-transcriptional regulating the actions are very important of cereblon law. But there is some studies suggest the CRBN can also binding to some part of DNA. So that is another some field. Yeah.
Okay. CRBN itself is an enzyme. So is there any some compensatory enzyme in the cells to fight to CRBN or inhibit the CRBN activity?
Yeah, I thought that there is some alternative role, some another different types of the proteins which can the working or compete with the CRBN because CRBN whole body knockout mouse still very healthy if that is if there is no some competitive or alternative proteins, maybe they can show there may be very severe phenotypes. So some of other studies suggest that this UPS system has many different players. So the different players have different targets. So what do you think is that maybe CRBN has a good role or bad role, but some of the metabolic stress, maybe CRBN have the increase of the CRBN have some bad role. Yeah.
Okay, thank you.
So is there any persons who make a question to the Professor Kim?
My name is Xiang Wu. I'm from Yanbian University. I'm very interested in the first speaker, Williams.
Okay.
Yeah.
Yeah, please stay the stand up, and anybody who make a question to the Professor Hyung-Kyu Kim.
Question. I just very interested in the you just study hypertrophy. I don't know in the hypertension hypertrophy and another kind of HCM. What kind of difference? Did you any data difference hypertrophy and hypertension hypertrophy? Difference in the serotonin expression of work.
Yeah, that is very important question. If that is how the hypertensions are stimulation of the hypertrophy CRBN working. So why didn't I checking if that is any? Because we only checking the phenotype of the CRBN knockout mouse. But I thought that is very important question. If we challenging to this knockout mouse to the more hypertrophic stimulation, maybe we can find out some different response to the stress. But I thought maybe we can collaborate to that kinds of work.
Okay.
Thank you very much.
Please, Derek.
Thanks, Professor Kim, for that great talk.
Yes,
you
sir.
showed that cereblon levels are increased in heart failure.
Yes.
Just wanted to know what was the cause of that and which cell type is secreting the cereblon.
That what was the, I got the data from the patient image and he said it's the ventricular cardiomyocyte. So they got the biopsies from some treatment of the patient. And that is the mRNA level is from the ventricular myocyte.
Okay, thank you.
Of regulation of cereblon level, I'm not very sure, but when we challenge that animal model to the stress, like some doxorobitions or some the myocardial infarctions, we found the increase of the CRBN level, mRNA level, and protein level. So some kinds of stress can increase the CRBN level. Maybe CRBN is also some of the sensor of some stress to some cardiac toxicity or some the physical damage. Maybe if you see my data, the difference or protective effect of cereblon knockout is much higher in the doxorubicin in this heart failure model. So that means maybe the more oxidative stress or the mitochondrial toxicity, maybe CRBN can be more working on that kinds of stress than direct myocardial infarctions, some physical and short walking. So still, maybe it's not insufficient the the answer how the stress increase the CRBN, but we are looking to that kinds of what kinds of transcription factors can increase the CRBN under the stress. Maybe Hyo-soo Kim will give us some clue.
Thank you.
Yes.
So actually, from the viewpoint, the clinician,
Yes.
actually, to my best knowledge, is there are no drugs to handling the calcium to make prolong the patient survival.
Yes.
So I think it's one of your the wonderful therapeutic mechanism for the cerebron is to making some handling the calcium
Yes.
channel. So do you expect in the beneficial over the past in some calcium handling some drugs, and then as I mentioned that the cerebron induced some hypertrophied in the myocardium.
Yes,
Actually,
yes.
so it's the heart failure with preserved ejection fraction. So
Mhm.
one of the mechanism is to get the hypertrophied myocardium. So
Yes, yes.
did you some examine the diastolic function in your some models?
Thank you for the very important questions. The first one is that if these kinds of the enhanced calcium regulation can be beneficial role in the patient. I thought maybe if that is too low, the calcium channel, we don't want to enhance the calcium level to the supra level, but if that is the decrease the cardiac functions, maybe CRBN can be the target, because some of the data, previous data, the thalidomide, the treatment to the heart failure patient, because actually that is invented for the immunomodulatory drugs, that show that the beneficial effect in the heart failure patient, but they didn't know about the cerebral law in their treatment. So because these kinds of the energy metabolism, the aspect, and the immunomodulatory aspect, and then maybe CRBN-related calcium, the aspect, can working together, maybe that can cause a new some target. So not only the single target, but the multi-target therapy can be thinking about that way. The next question was that these kinds of the hypertrophy. So as I told you to the Chung, but we didn't yet test more hypertrophic stress to our model, but I thought these are very challenging, and then we should checking the kinds of aspect.
Okay, thank you.
Thank you
So
very much.
maybe please first.
Yes. Yeah, that is increased. Yeah. So, the two types of model, doxorubicin induced heart failure, and the myocardial infarction induced heart failure model. So we do not prolong this experiment about 1 week. We started, but that is gradually increase the CRBN level in the heart. Can be. So one of our project now, we started about the role of cerebral in the fibrosis. There is Dr. Ani is the very first professional, and then we got some help from the Yongsu. So I thought there's another role of CRBN is the regulator for the fibrosis, cardiac fibrosis. So that is maybe if I invited next year, I will give some talk about related fibrosis or metabolic cardiomyopathy related study. Thank you very much.
Yeah. William, please stand up to the microphone. Yeah. Chen, maybe you want to make some question? Okay.
As I very interesting. I just wonder, and I don't know, now it's very popular the single cell lysis. I know you studied about different place in the order. So Andrew just use the compound 21 is treatment. Which kind of cell? For example, just focus in the brown adipocyte and white type cell where worked in the expression as work to the compound 21.
Yeah, so
Oh, we can clearly
Okay, sorry,
see your
sorry.
voice.
Okay, so the whole purpose was to see if we can transform the white adipose tissue that's very prevalent in the abdominal aorta, as I showed you, to more of the beige type, because that's the area that is way more prone to atherosclerosis as well. You look at the thoracic aorta, I'm sure you know this very well, there's very little atherosclerosis in the thoracic aorta. And part of that might be because of the brown fat adipose tissue that's surrounding the thoracic part versus the abdominal part. So another thing that we're doing right now is we're trying to implant, actually, the brown adipose tissue, because in mice, it's very easy to do, into the parts that are covered with white adipose tissue to see if we can modulate the atherogenesis that way.
Okay, thank you.
Okay, please. Please on.
Thank you. First of all, thank you so much for your great speak. And I have just one question. Is you just said that compound 21 is a very potential drug for treatment for hypertension. So the angiotensin 2 receptor AT1R and AT1-2 probably have the opposite effect.
Yes.
It's AT1R is the contractile vessel, and the other one is relaxed vessel.
Right.
So in clinically, traditional treatment for hypertension is the inhibitor for the AT1R. So compound 21 is an agonist for the 22R, right?
AT2R, yes.
AT2R. Yes. So my question is, what is the advantage of the AT2R agonist compared with the AT1 inhibitor?
Okay, so the AT2R is expressed in the pathogenic conditions, as I mentioned. So that's what we're targeting. You can't really target AT1R because that's ubiquitously produced or expressed in all of the vessels. So the reason why we're targeting AT2R is because aside from all other factors that might affect the expression of AT2R and other pathogenic conditions, if you just isolate the atherosclerotic condition, it is expressed in that sense. So that's what we're targeting. There are many studies to do to see if there are other effects, of course, but right now we're just focusing on the advanced atherosclerotic formation.
Thank you.
Okay, Yongso, please.
I have a quick question.
Sure.
So did you compare the circulating adipokine profiles in your mouse models?
No, we haven't done that yet. That's coming. As I said, it's a preliminary results that I gave you today. It's all being done right now in Spain, actually. Yes.
Okay, please.
I have a quick question because a few years ago we have done with isolated ventricular myocytes and applied angiotensin 2, which induced angiotensin type 2 receptor. And the downstream of this signaling is neuronal type of nitric oxide synthase, protein induction, and then activity. So my first question is, do you think this is due to the nNOS in your model? And the second question is, just like Professor Chung says, that we would like to just extend this study to peripheral, the ventricular perivascular fats, and then effect in the myocardium. So we're struggling to find which type of cells to focus on. Like after your talk, maybe endothelial cells of the coronary arteries, or maybe the adipocytes from the perivascular fats, or maybe just the ventricular cells. So which focus do you think is the most for this study?
So the focus that we had was on the endothelial cells.
Okay.
So that's the focus. The reason we chose the aorta is because it's an easy vessel to look at, and because of the differential expression of the different adipose tissue types on it. The coronary vessel, obviously, is the most important for humans. We checked for perivascular or arterial in this case,
Okay.
fat in the coronary arteries of mice, but we couldn't find it because it's just too small.
Oh.
And the way the vessels are shaped in a mouse is very different than in humans. So that's why we're using that. But the real focus is on the endothelial cells. Yeah, that's what
But
we're focusing on.
presumably, I think it is the enos, not enos.
Yes, most likely, yeah.
Okay.
So that's, well, part of it anyway. Yeah. Thank you.
So, like actually, it's A2 type 1 receptor antagonist with the type 2 agonist can make some synergistic effect on the cardiovascular disease.
I'm sorry.
A2 type 1 receptor antagonist and the type 2 receptor agonist can make some synergistic effect on the cardioprotective effect.
Maybe. You would probably know as a cardiologist more than I do whether that's actually the case or not.
Okay, so would you like to introduce some second speaker, Hyosu Kim? Is there any questions or comment to the Hyosu Kim?
Question for you.
Okay.
Yeah. I cannot understand what you're saying. Do you comment more detail regarding the expression of AT2 receptor? You just mentioned AT2 receptor are expressed in certain cells under abnormal condition. Is it right?
No, not in adults.
Not in adults.
Yeah, only in fetuses.
So which kind of cells express AT2 receptor in adulthood?
Endothelial cells. So endothelial cells.
Normal endothelial cells
Yeah,
as well
I'm
as
looking at atherosclerotic plaque only, right? I'm not looking at other pathogenic conditions.
endothelial cells, a normal vessel does not express AT2 receptor.
Right.
Okay, only endothelial cells from abnormal vessels express 2 receptor.
Of the tissues that we looked at, yes. If you look at the atheroma, the endothelial cells are robustly expressing AT2R, but not in the normal.
Did you decipher the mechanism why endothelial cells in abnormal vessels express 2 receptor?
No, not. We haven't looked at the mechanism.
Okay.
Yeah.
So, okay, please, Chen.
Yeah, thank you, Professor Kim. Yeah, very interested in your study. I just wonder one thing very interesting because we are so the knockout mice, they know that ADPT knockout very quite different, the reduction in the several pipa gamma, pipa alpha of something. But however, some knockout mice maybe compensate also something very higher is expression some maybe is molecular. How about your study just focus only down-regulate another gene, no up-regulated gene in the knockout?
We analyzed transcriptome between wild-type embryo versus homo knockout embryo and see several gene clusters. But we focus on, there are numerous gene clusters. We focus on first gene that control the mitochondrial biogenesis and that control the junctional proteins. And regarding the mitochondrial controller, there are some genes increased, but PGH1 alpha really is shut down. Thus, we guess there may be some conflicting gene effect. Some genes, positive genes, are increased, but negative, some genes may activate, but reduction of PGH1 alpha was so profound that may exert the main results of mitochondriopathy. Regarding the cell junction protein, all the junctional proteins are suppressed because most of them are under the regulation of P38. That's the reason why we can rescue the phenotype by anisomycin.
Sujin, okay. Professor, please.
Thank you very much for your wonderful speak. And I have 2 questions actually. First is this is cardiomyocyte induced from the IPS via the latrophilin 2 is beating automatically in vitro?
When we induce the differentiation of iPS cells toward CMC, they transiently cardiac progenitor cells emerge. Then they express latrophilin 2. They finally differentiate to mature cardiomyocytes and beating. They maintain the latrophilin 2 expression. Thus, latrophilin 2 expression starts in cardiac progenitor cell stage and all continues. Thus, in normal heart, myocyte express latrophilin 2 very strongly.
So actually, now we usually use the WNT induction kit to induce the cardiomyocytes from the IPS. So this is latrophilin 2 is the underlying pathway of the WNT signaling pathway. Is underlying the pathway of the WNT signaling.
WT signaling?
Yes.
What does it mean?
It's like more recent research about IPS-induced cardiomyocyte. We used the WNT induction kit. It's like a very, it's like a signaling pathway. The first 3 days we activate the WNT pathway, and after 3 days when we inhibit the WNT pathway to induce the cardiomyocyte from the IPS. So what is I don't know the laparoscopic.
WT means wind signaling?
Yes, wind signaling. Yes, wind signaling.
Okay, in our second paper, we decipher the level and mechanism of latrophilin 2 to determine the cardiomyocyte differentiation. There are several genes involved in the first heart field and several genes second heart field. There's first heart field may contribute to development of left ventricle, second heart field and right ventricle and outflow tract. When we block the latrophilin 2, all the genes involved in first and second heart field shut down. Thus, we guess latrophilin 2 is really the top hierarchy genes that controlled NKX and many MAP2C, GATA4, all the genes are shut down. I guess upstream of latrophilin 2 may be MSP1, that is initial genes to induce commitment toward the mesodome. And then latrophilin 2, and then many other TBX5, GATA4, MAP2C, NKX. Thus, we reported such kind of hierarchy of genes that are involved in cardiac differentiation.
Final question.
Professor Song, I think it's okay.
Sorry.
The short question because time is up, and then maybe some 20 minutes in the reception will be held in the room A. So please.
Sorry. Final question. The cardiomyocyte induced by the latrophilin 2 is more like the atrial cardiac myocyte or ventricular cardiac myocyte?
We are now doing transdifferentiation of fibroblast to cardiomyocyte by single gene transfection latrophilin 2. Now we are analyzing the phenotype of induced CMC that are more likely to atrial cardiomyocyte. We compared the transdifferentiation efficiency between latrophilin 2 single gene and GMT triple gene. Dr. Yeda are now trying to make high efficiency of induced CMC by triple gene, GATA4, MAP2C, and TBX5. They use triple gene, GMT. We use single gene, latrophilin 2. Efficiency is about 15%, same. That is another supporting evidence. Latrophilin 2 is a kind of master gene. Single gene would be enough to the triple gene downstream.
Great. Okay, thank you very much, Professor Kim. We have to draw the session to a close now. I'd like to thank all the speakers for their very interesting and excellent talks. Also thank the panelists for their questions and the audience. And I hope you enjoyed the session and look forward to the rest of the meeting. Thank you, everyone.